can see for working on biomarkers, we are trying to use these cancer biomarkers in lung cancer in order to help us to achieve better diagnosis, treatment, and prognostication, and if possible in future, to help monitor the disease, as I'm going to show you in a moment. And talking about lung cancer, the most common one, the most common histological subtype is still non-small cell lung cancer. And among non-small cell lung cancer, we have adenocarcinoma and squamous cell carcinoma, and you can see a lot of the biomarkers or so-called oncogenic driver mutations has almost been defined. Of course, there could be new ones to develop, but we now know a lot, and the study of the biomarkers hopefully will lead to the practice of precision medicine in the future. And by practice of precision medicine, we are trying to identify the management approaches that will be effective for patients based on the genetic information and also the other characteristics of the disease. And you can see from this table that we now have different therapeutic options for advanced stage non-small cell lung cancer, and based on the available oncogenic driver mutations, that we can detect, namely EGFR, ALK, and also on resistance to detection of T790M. And we have different approved therapy based on the biomarkers that we can detect in the tumors or in other clinical specimens. And we not only are we based on the presence of oncogenic driver mutations, but we are also basing on the histology of the tumor to guide our subsequent choice of therapy, whether we will use specific targeted therapy or we will use chemotherapy in combination of the VEGF inhibitor. And nowadays we also have the so-called immunotherapy, as I'm going to introduce to you later. So in our population, we are facing with a major proportion of patients who have EGFR mutation, no matter they are smokers or non-smokers, although in this proportion, a lot of them are non-smoker. And EGFR is the most common one. And I'm sure this is the same situation in Taiwan here. And now it has been established that EGFR mutation, if detected, is a biomarker. But not all the mutations are the same. And some of them are known to be sensitizing to EGFR TKI, especially deletion 19, L858R, or L861Q in exon 21. Most of the times, these mutations are located in exon 19 and 21. And if it happens that there is another mutation in either exon 18 or 20, it usually implies partial sensitivity or even resistance to EGFR TKI. But nowadays, fortunately, there is the so-called development of a third generation EGFR TKI that can target this EGFR mutation if such resistance happens. So the EGFR mutation, if detected, will guide the choice of the use of a first-line therapy with EGFR TKI in stage 4 non-small cell lung cancer. This has been established, and this has been the use of EGFR TKI first generation in the patients bearing EGFR mutation has been translated into survival benefits. So the correct targets being identified will give will guide us to the correct drug. And not only is the types of mutation that is detected, but it's also the fine type, the subtype of mutation. And for example, the one of the mutation that is more sensitive to afatinib, so-called second generation TKI, that will translate into survival benefits if the patients bear the exon 19 deletion. So it seems that the biomarkers has been established, but there is still more to know, especially with regard to the EGFR mutation. And we have tried previously to profile the expression, the gene expression of the tumors bearing different kinds of mutation with respect to sensitizing mutation and also so-called less sensitive mutation. And it seems that certain genes could actually be involved in such mechanism. And one of the genes that we have explored is the LSS2, and it is supposed to be a tumor suppressor gene that is present if it is in high level, will protect the patient or protect or the tumor against progression. That means the presence of which will suppress tumor growth. So, and we have found that the expression of the LSS2 in the tumor is associated with survival outcome, so that the patients with higher expression of this LSS2, a tumor suppressor gene, that means when it's expressed, it will help to suppress the tumor, is associated with a better survival in patients. And not only is it happens alone, but if it is detected concomitantly with EGFR mutation, it seems that the EGFR mutant with high expression of LSS2 always fare better than those EGFR wild-type patients who have low LSS2 expression. So we do not know if the two genes are actually sharing some of the downstream pathway or working together to promote tumor growth or suppress tumor growth. And we try to explore further with the patients or the tumors and the cell lines with high LSS2 expression. We try to knock them down, stop their expression. And it seems that after stopping the expression in those highly expressed LSS2 tumors, there will be lesser invasion of the tumor cells after the LSS2 has been knocked down. We are working on further to see if we can work the other way. For example, to knock the LSS2 up in those patients, in those tumors or cell lines that have low LSS2 expression, to we express them to see if there is any further increase in or promotion in tumor regenesis. Hopefully, we will have some more results to share with you in future. So after EGFR, we have another one that is the ALK. ALK is another biomarkers. We used to believe that the ALK and EGFR won't happen together in the same patient, which is usually the major story. These driver mutation usually will be present in a mutually exclusive way in one patient, so that if you have one detected, for example, EGFR mutation detected in one patient, usually you won't find others. But so, for example, if we have another type of patients within the EGFR wild type that you cannot detect EGFR mutation, a lot of times, sometimes, in a rare population, you may find patients with the ALK rearrangement. And this ALK is not gene mutation. It's based on gene translocation. That means the gene actually is present on a chromosomal location, and as a result of translocation, it's being moved to another location in another location of the chromosome, being bind with another partners, and thus revealing its oncogenic potential, and hence our use as a biomarker for detection. And this ALK translocation has been known to be present again more commonly in patients with adenocarcinoma, never smokers, and they're relatively younger than among patients with lung cancer, although we do not see much gender differences. And you can see it's not gene mutation. We are not counting the nucleotide, the APGC in the DNA, but this time is being detected as chromosomal rearrangement, that if we tag the chromosomal location and the gene with one probe, when the genes separate, it becomes translocated, the original gene will get separated. The probe that is got detected in this, what we call fluorescence in situ hybridization, will got separated if this is translocation positive. You can see the separation of the red and the green probe. The red and the green probe over here, over here. If there is no translocation, a lot of the times they will be binding together. Okay, and but the scoring of this is difficult. As you can see,